we expect the cells of the ectoderm to manifest morphological changes in order for them to facilitate the process of neurulation. So before we take account of these cellular changes, let's first describe neurulation as um, two major events that combine together and later on produce a consummated effect. Right? So, two major events of neurulation in majority of vertebrate animals are primary neurulation and secondary neurulation, which actually differ in terms of cell behavior and tissue and cell reorganization. In primary neurulation, primary neural induction, there are a variety of stages that we can appreciate, from neural plate thickening to the closure of the neural groove, giving rise to the neural tube. So these stages are defined as formation of the neural plate and shaping of the neural plate cells, also known as primary induction. In this event, we expect the inductive factor from the notochord to be continuously expressed by notochordal cells, by notochordal genes. This inductive signal being sent outward or towards the dorsal side of the animal, towards the responding tissue that will later on become the neural tube, right? After which, after primary induction, we expect the neural plate to undergo bending. And in this bending, this bending, identified parts of the thickened neural plate will undergo hinge formations. Two hinges or two hinge points can be identified, known as DHP or DLHP, dorsolateral hinge points, and MHP, median hinge points. As bending continues, the thickened and elongated neural plate cells to form a groove, right? Uh, Sides or ridges of the groove will later on meet, right? Closing the groove or forming a continuous closed tube known as the neural tube. So let's take a look at the cellular changes. So here we're seeing on the first panel a picture, representation of the genital ectoderm that is not yet divided. So let's say, for example, that chorda mesodermal notochordal cells have started the development or just started the development of notochord tissue underlying the ectoderm. Okay, expect the inductive factor to be produced by these cells. Inductive factor later on will be interpreted by competent ectodermal cells, and that will produce a change in the morphology of these responding ectodermal cells. From flattened cells, they will later transform as elongated cells by the virtue or by the help of microtubules. Their microtubules help them elongate, right, along the vertical axis. So the cells of the neural plate, particularly the cells on the middle, will elongate, right? And later on, microfilaments on the dorsal side of the cells, as we are seeing here in number 2, microfilaments on the dorsal side of the central cells will contract, forming them or deforming them as wedge-shaped aggregates of cells. Okay, cell wedging will allow the formation of a groove, the neural groove. You can see here the depression, right? So, which is resulting from the formation of this part known as the median hinge point. The median hinge point will allow the sinking of the neural groove towards the coelomic cavity of the animal, and then later on, this wedging on the middle will also produce wedging on the sides.